Good morning, students. Let us continue with the chapter number 14, fermentation. We have studied that there are two ways of respiration, anaerobic and aerobic. For anaerobic respiration, organisms undergo fermentation. Let us study two types of fermentation in more detail. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by sets of reaction where pyruvic acid is converted to carbon dioxide and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organisms like bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure. In animal cells also, like muscles during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the processes. In both lactic acid and alcohol fermentation, not much energy is released because it is incomplete oxidation. Less than 7% of the energy in glucose is released and not of all it's trapped as high energy bond of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is net ATPs? That is synthesized. Calculate how many ATP are synthesized and deduct the number of ATP utilized when one molecule of glucose is fermented to alcohol or lactic acid. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to the synthesize a larger number of ATP molecules needed for cellular metabolism? Let us see how fermentation takes place. Glucose, when converted to the glyceraldehyde phosphate, phosphoglyceric acid and pyruvic acid, then pyruvic acid goes to the change in either lactic acid or ethanol. In both the cases, CO2 is removed and NADH is oxidized to NAD+. Very less amount of energy is released during this conversion, and due to incomplete oxidation, energy released is not sufficient. Electron transport system and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to oxygen, resulting in the formation of water. The metabolic pathway through which the electron passes from one carrier to another is called electron transport system, present inside the membrane of the mitochondria. Electrons from the NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by NADH dehydrogenase that will form complex 1, and the electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalence via FADH2 complex 2, that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinone to ubiquinol is then oxidized with the transfer of electrons to cytochrome C via cytochrome Bc1 complex, that is complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for transfer of electron between complex 3 and 4. Complex 4 refers to the cytochrome C oxidase complex containing cytochrome A and A3 and 2 copper centers. When the electrons pass from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase, that is complex 5, for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH give rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to terminal stage of process. Yet, the presence of oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor. Unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation, in respiration it is energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane-linked ATP synthesis as explained by the chemiosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase complex 5. Let us have a look on the ETS. From the complex 1, NADH dehydrogenase, NADH is oxidized to NAD. The electrons are transferred to the FMN, FES and then to ubiquinol to reduce it to ubiquinol. Same here in the complex 2, purple key-like structure. There, FADH2 releases the electron and again it reduces ubiquinone to ubiquinol. After that, the electrons move to the complex 3, then to complex 4 via cytochrome C. Complex 4 is made up of cytochrome A and A3 along with the 2 copper centers, after which the electrons are passed on to the complex 5 that is made up of ATP synthase. ATP synthase is made up of 2 particles F0 and F1. The F1 headpiece is a peripheral membrane protein complex and contains the site of synthesis of ATP from ADP and inorganic phosphate. F0 is the integral membrane protein complex that forms the channel through which proton cross the inner membrane. The passage of proton through the channel is coupled to the catalytic site of F1 component of the production of ATP. For each ATP produced, 2 protons are passed through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. Let us have a look on the complex 5 that is made up of F0 and F1. F0 is embedded in the membrane while F1 is protruded out towards the matrix. Whenever the protons cross through the F0 F1 particle, the ATP synthase get activated and results in the synthesis of ATP by phosphorylation of ADP. Let us see the respiratory balance sheet. It is possible to make calculations of the net gain of ATP for every glucose molecule oxidized. But in reality, this can remain only a theoretical exercise. These calculations can be made only on a certain assumption. That first assumption, this is a sequential orderly pathway functioning with one substrate forming the next and with the glycolysis, TCA cycle and ETS pathway following one after another. Second assumption, that NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. Third assumption, none of the intermediate in the pathway are utilized to synthesize any other compound and they just follow the single pathway. Fourth assumption, only glucose is being respired. No other alternative substrates are entering in the pathway at any of the intermediary stages. But this kind of assumptions are not really valid in the living system. All pathways work simultaneously and do not take place one after another. हमें ये याद रखना है कि लिविंग सेल जो है वो एक ट्रैफिक रोड की तरह है. जैसे ट्रैफिक सिग्नल्स पे 4 तरफ रोड जाते हैं, द सेम वे कोई भी कंपाउंड एक से ज़्यादा पाथवेज में लिंक्ड हो सकता है. Substrates enter the pathways and are withdrawn from it, and when necessary ATP is utilized and synthesized also. Enzymatic rates are controlled by multiple means, yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system. In extraction and storing energy, hence, there can be net gain of 38 ATP molecules during aerobic respiration of 1 molecule of glucose. Now, let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of the glucose, whereas in aerobic respiration, it is completely degraded to carbon dioxide and water. In fermentation, there is net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions. NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration.